So my talk is going to talk about something that is a review. It's a review because the new science that I'm talking about is now an established science, and it all has to do with the nature of DNA. When I was teaching in the medical school, I was teaching the concept that DNA was the molecule that controls your life. It programs you. It's part of your inheritance that shapes and determines your future. And this is what I was teaching in medical school, that you are an expression of your genes. Your genes control who you are. And in that understanding, it was really emphasized by the work of Francis Crick, who was a co-partner with Jim Watson in understanding the DNA double helix. And all of you have read this understanding in your textbooks. It's called the central dogma. And it's interesting because it's still in the textbooks today. And this was some work by Francis Crick. And it was a story that information in biology flows from the DNA to the RNA and then into the protein, and that this is a unidirectional flow. And you say, well, why is all this relevant? And the answer is, you are the protein. You are the expression. Well, why is that relevant? If information can only come from the DNA down to the protein, then by definition, then you cannot influence your DNA because the information doesn't go back up the other way. Understanding this central dogma by Crick shaped the whole medical establishment to say you are just a consequence of your genes and you cannot really go back and influence these genes. Well, what's most interesting, while it's still in the textbooks at this very moment, is this fact that most people haven't ever realized. The central dogma was a hypothesis. That's all it was. It was never scientifically studied. It was never even, observed to find out if it was true or not. Everybody just believed it because they were looking for the answer. So when it came out in 1958, it was an idea. And then by 1968, it was in all the textbooks. Then it's 1978. Then it's 1988. And guess what? Nobody remembered that it wasn't even a scientific fact. It was just a hypothesis, an idea. And we bought into it that it's still in the textbooks today. Well, the significance about this is that when we buy into this belief, we buy into the belief that genes control your life and that you, as a protein element, cannot go back and change these genes. What was most interesting for me was I never even looked up the word dogma, although I'd been teaching it for 10 years, until I left the university. And that was the biggest shock of my life because the definition of dogma is a belief based on religious persuasion and not scientific fact. I had been teaching religion in medical school for all those years. And there's still dogma people that are still following that belief. But the issue is this true? Well, it's so true that to medical people in the conventional world, not to the medical doctors, but to those people who define and devise the curriculum, they created a model of what is conventional medicine, and the medical model is simply this. You are a biochemical machine controlled by genes, and this is what we operate from. And so starting with the genes as control, then we're led to this understanding as on the cover of a Life magazine, where you're born that way. Meaning, when they started looking at the role of genes, it wasn't just now the physical structure, but they started to assign to genes things like function and emotions and the behavior. And all of a sudden, all the genes were controlling every aspect of it. It was so interesting because everybody started looking at every emotion and character and trying to identify the gene that found it. And I even remember there was a guy who did a study on the happy gene. He was looking for the happy gene. How do you find the happy gene? Well, you get a bunch of sad people, and then you get a bunch of happy people, and you look at the genes of both, and guess what? He came up and he said, here's the gene for happiness. And it was like, what the hell does all that mean? It means this. It means that what happens if you're a recipient of a sucky happy gene? Then basically represents is your whole life. There's nothing you can do about it. Why? Because you can't control the genes. Of course, all that's all a sham, because if you anticipate you're looking for a gene and then you do science with a predetermined end, then you find your determined end, but guess what? It's no longer science. You already concluded what you were looking for, and then you start looking for it. So it's really out of the phase of science. But why is this relevant? And the relevance is this. I tell you genes control your life, and then you say, well, where did I get my genes from? I said, well, at the moment of conception, your father and mother donated each a half a set of chromosomes to give you a full set, and you didn't have anything to do with picking those genes as far as we know. And then I go, okay, wait, you don't like your traits? You say, oh, but I can't change my genes either. And all of a sudden you start to realize that your life is not under your control, it's under the control of your genes. Well, that makes you a victim. And we are programming people to be victims because we say your heredity controls your life and you have no control. And I say, well, what's relevant? And the whole most important part is if I acknowledge that you're a victim, then you must acknowledge that you need a rescuer because you can't do it yourself. And all of a sudden, we give over all our power over our life to the so-called rescuers, which is the pharmaceutical industry, which is there. And all of a sudden, we realize, oh, my God, you know, we're just so weak and frail and vulnerable. There's nothing we can do. And that's where we live from day to day. So when people look at their lives, other than people like Evan, they look at their lives and say, oh, can't do anything about it. Just got to live with this stuff. And the fact is this really true? So I say, okay, look. Here's something about the fact that the genes control life, and the genes are primarily all in a structure called the nucleus. And that's why even today in the textbooks, they talk about the nucleus as the brain of the cell. And you say, well, why is the nucleus the brain? The answer is very simple. The nucleus has the DNA, and the DNA controls life. So the nucleus is the collection of all this control. I say, well, if this is true, then let's understand something about the brain. If you have an organism, and you remove the brain from the organism, what is the consequence of that? And the answer is... the organism dies. That's what is automatically the consequence. And I say, well, what if I go into a cell, remove the brain of the cell? It's called the nucleus. It's a process called enucleation. And in fact, I did this enucleation research. I was doing that 47 years ago. And at that time, what did I find out? What did I know for 47 years about this fact? And the fact was this. You can remove the nucleus from the cell, but guess what? The cell lives and its behavior is totally unaffected. Cells can live for 2 or more months with no genes in it at all. And you say, well, how does this happen? The answer is basically, number one, the belief system that the nucleus is the brain of the cell is a totally flawed concept. Because a cell can live without the genes for months, and all of a sudden we have to say, then we have a complete misunderstanding about how a cell is controlled. But I will give you now this fact, and this is a hard fact. There's no quibbling about this fact, and it's simply this. Genes do not control biology. And there's a very simple and fundamental understanding as to why that's true. And the answer it comes about is, what is a gene? A gene is a physical blueprint to make a protein. That's what a gene is. I said, well, why is that important? I said, because it's not like a blueprint, it is a blueprint. I go, well, what does it mean? I go into an architect's office. She's working on a blueprint. You lean over her shoulder and you go, excuse me, is your blueprint on or off? And she looks at you and goes, what are you talking about? There's no on and off to a blueprint. Precisely. Why is that relevant? Because we've all been programmed that genes turn on, genes turn off, genes control you, and the fact is this, it's completely false. There is no on and off to a gene because it's a blueprint. And all of a sudden you have to say, but all the years that we were talking about a gene caused cancer, a gene causes things, it's like genes cause nothing. Genes are just blueprints to make the building blocks. And when we get to that, we can start to take our mind off the consequence of genes controlling our lives. Yeah, but then all of a sudden I say, well, go back to the medical model. This is what we operate from, biochemical machine controlled by genes. It goes, this is totally wrong. And this all of a sudden says, well, then it makes sense why even the Journal of the American Medical Association has reported that medicine is one of the leading causes of death in the United States. And the principle is completely founded on the wrong concept. And this is why it's failed, because they keep going back to the source of the genes. That's where the problem is, and that's not where it is. So I really want to start with just giving you the basic understanding of life. I want to talk about the primal element of life. And the primal element of life is not the DNA at all. The primal element of life are proteins. Your body, your cells are made out of about 150,000 proteins. Primal element? Yeah, that's exactly what the Latin protein means, primary particle. So the primary element of life is protein. And I said, well, how does life work? And so here it is, the most interesting and shortest little story about how life works. It's so simple. It goes like this. Proteins are building blocks that create this body. Fine. But then here's the most important other understanding. Proteins, 150,000 different ones, not only have a physical structure, but protein molecules are unique because they can change their shape. And why is that important? Because when the protein responds to a signal from the environment, the protein undergoes what is called a conformational change. So, for example, an environmental signal can alter each of these proteins and cause them to change shape, which causes them to move. The movement is used by the cell to do work. And all of a sudden you say, well, where does life come from? The answer is from proteins responding to environmental signals. So bottom line, that's the simplest understanding of life. You're a protein machine and you respond to environmental signals. So the significance about all this is, how does life work? The magic is movement, and the magic comes from proteins changing shape. Well, all of that was going on in my research. Back 45 years ago, I was cloning stem cells. And what's interesting about stem cells is that they're embryonic cells that are in your body right now. If you are here, then that's a clear proof that you have stem cells for a very simple reason. Every day you lose, in Carl Sagan's words, hundreds of billions of cells are dying every day, and you have to replace them, because if you don't replace them, how many days can you go before you start to die? And so the significance is, every day you're losing those hundreds of billions of cells, and every day you're making new hundreds of billions of cells, and they come from what are these stem cells, embryonic cells that are scattered throughout your body right now. My research back in 1967 was to take one stem cell, put it in a petri dish all by itself, and then it divides every 10 or 12 hours. And in a week, I have 50,000 cells in the petri dish. Well, what's significant about this is that the 50,000 cells are all derived from one cell. So the significance is this. I have 50,000 genetically identical cells in the petri dish. Now comes the experiment. What I do is I split the cells up into 3 different petri dishes, and I change the environment in each petri dish. And I say, what's the environment? I say, well, cells are like fish. They have to live in fluid. So I create a fluid thing called a culture medium, and the culture medium is what I use to grow the cells in. And I said, well, what defines a culture medium? Where do you get that from? I say, we try to replicate the conditions of the internal fluids of the body, like the blood, and create the equivalent of blood so that the cells can get all the nutrients and information they need to grow. So what did I do? I put these cells into different dishes, but I changed the culture medium chemistry a little bit in each dish. So in the first dish, my stem cells go in there, and in culture medium A, environment A, they form muscle. I go, cool. Then I say, take some more cells out of that stem cell dish, put it in a second petri dish with a different environment, environment B, and they form bone. And then lastly, go back to the dish, take out some more cells, put it in a third petri dish with a different environment, and they form fat cells. Now, you don't have to be a rocket scientist to answer this question, and it is profound. What controls the fate of the cells? And the answer is the environment, because they all were genetically identical. So at some point, we have to start recognizing that the environment is providing information which is involved with selecting the gene activity. Well, how does all this come about? Well, in studying my cells in tissue culture, what I started to recognize was this. As I changed the chemistry in the dish, the chemistry rained down on my cells in the petri dish. Some of that new chemistry attaches to the skin of the cell, the membrane of the cell. These signals picked up by the membrane of the cell are then relayed by the membrane into the cell, where those signals affect the proteins. And remember why? Signal plus protein creates action. So all of our functions, respiration, digestion, excretion, they're all due to proteins moving, but all proteins move because of signals. So the signal on the surface of the cell, the skin, is relayed inside. And it affects the proteins, which then adjust behavior because the movement of the proteins creates behavior. And if you don't have the necessary proteins for that behavior, the signal then goes into the nucleus and is involved with selecting the genes to make those necessary proteins. Well, all of a sudden, the whole idea of a gene-focused biology shifts over to the membrane. And it starts to recognize that the membrane is the controlling factor of the cell. And so the significance of that is that there's two new sciences, actually one new field of science with two subdivisions. It's called signal transduction. And instead of focusing on the genes now, we're looking at what about those signals that are controlling things? Signal transduction means the science of how signals are translated into biology. Well, those signals affect the proteins of the cell and affect the behavior. But some of those signals go into the nucleus and are involved with affecting the genes. So a part of signal transduction where the signals affect heredity is a new science called epigenetics. And the significance of epigenetics, it is a revolution that changes the world. It is the most important revolutionary discovery in biology, tantamount to the finding of quantum physics as opposed to classical Newtonian physics in the world of physics. Epigenetics is a game changer. It changes everything we know for this reason. Epi means above. When I say skin in the Latin terms, it's called epidermis. What does that mean? The layer above the dermis. So when I say epigenetics, what does that mean? I say, well, wait, what have you been programmed with? Genetic control. Control by genes. I say, what's the new science? It's called epigenetic control. I say, what the heck does that mean? I go, control above the genes. And all of a sudden you start to realize this, the control wasn't in the genes ever at all. It was above the genes, and it comes from the environment. And it turns out that the skin of the cell, which is translating the environmental signals, is the brain of the cell, and that's why we call it the membrane of the cell. And cells are miniature people. So a lot of people say, well, if cells are miniature people, then are you telling me, Bruce, that my nervous system is connected to my skin, my membrane? The answer is absolutely. If you took embryology, you would know that because the nervous system is derived from one of the germ layers in the fertilized egg called the ectoderm. And there's only two structures that come from ectoderm, skin and nervous system. So you and the cell are exactly the same. So a cell is a miniature human. And then when you have signals hit your surface, your skin picks up the information, translates it via the nervous system, and controls your biology. So I say, yeah, how do you do that? Well, you have things called receptors. Eyes, ears, nose, taste, touch, pain, temperature. All these receptors are built into your skin. That's how you read the environment and how you make a response to that environment. So in my studies, I wanted to find out how does that environmental signal ultimately control biology. So I went into the membrane, started to look at the structure of the membrane, and found something like this. These are proteins built into the membrane. So it's like a little cookie cutter cutting out a piece of membrane. There are two classes of proteins that I need to talk about. Receptors, which are proteins with antennas on them, which is the cell's equivalent of eyes, ears, nose, taste, touch. This is receiving environmental signals. But once you have the signal, you have to convert it into a biological response, and that's the function of the second protein called the effector. So remember, environmental signals change the shape of the protein. So how does this work? Well, what we see is we have the receptor protein with the antenna, the effector protein, but there's a new protein involved, the green one, which is called the G protein, processor protein. It's needed to connect the two. But look, the shape of the receptor and the processor don't fit, but the signal from the environment shows up. And when the signal from the environment shows up, then what happens is, oh, I'm going to have to start that again. I'm so sorry. The PowerPoint, Bill Gates, this thing didn't work right there. But the whole idea, I won't push the button now, but I'll let you show you. The shape of the processor and the shape of the receptor don't match, okay? And so now what happens is this. An environmental signal affects the protein, changes the shape of the protein conformation. Now a processor can connect the receptor to the effector, and the effector sends a signal into the cell, and when the signal of the environment goes away, the switch is broken. So it's a switch. And there are 100,000 switches on the surface of the cell responding to 100,000 bits of information coming from the environment. So the whole nature is all of us have switches built into us to read an environmental signal and then make an appropriate response. So the primary signal is picked up by the receptor, and the secondary signal that goes into the cell is what controls the behavior of the proteins and of the heredity. So how does that really work? Well, this is a picture out of a new textbook which is involved with signal transduction. And you can see the protein as little colored blobs in there and the membrane over the top. And I say, how does it work? And I say, well, a signal from the environment affects the receptor and then sends signals into the cell. And then as a result, the cell's proteins move in response to environmental signal. That's called signal transduction. So the signal comes in, the signal goes through the proteins, pass from one protein to another, passing the signals, and every time you pass a signal to a protein, it creates behavior. So simple point, signal causes behavior. No signal, no behavior. And that's called signal transduction. But I say, but sometimes the signals go into the cytoplasm and then into the nucleus. And into the nucleus, the signals affect the readout of the genes, and this is controlling gene activity. So again, signals from the environment control gene activity. And this is the new science that's called epigenetic control, how environmental signals go in and control that. I said, well, how does that work? What's going on in the nucleus, how a signal does that? So understand that, we have to take a nucleus and open it up. When we open it up, we find 46 chromosomes, 23 from your mother, 23 from your father. The chromosomes are the hereditary units. I say, well, what are these chromosomes? What are they made out of? I say, the chromosome is 50% DNA and 50% protein. I say, well, what's the DNA? I already told you, the DNA is the blueprint. And all of a sudden the question is, what's the protein? Well, for 50 years, nobody gave a damn about the protein. What they wanted to do was study the genes. So what would they do? Isolate the chromosome, separate the DNA from the protein, save the DNA for genetic research, and throw away the protein. For 50 years, they threw away 50% of the nucleus until somebody said, maybe that protein is doing something. And that's when we started to find out, what the heck is this protein doing? So basically, here's how it works. There's a chromosome, and the chromosome is like my arm with a sleeve, and the sleeve is protein, and underneath is the DNA strand. And so if you want to read the genes, you have to do what? What did I do to show you the DNA? I had to remove the sleeve. Well, the sleeve is the protein. Yeah, and I go, yeah, but what influences a protein to change shape so it pops off the DNA? And the answer goes right back to the signal. So how does it really work? Signals come in from the environment, pass in the nucleus, epigenetic, the signal binds to a chromosomal protein. It causes the proteins to change shape, and when the protein changes shape, it detaches from the DNA, and all of a sudden, voila, the DNA blueprint's exposed. And I go, oh, now what do we do? I say, well, you want to make a copy of the blueprint. And that's called RNA. It's like a Xerox copy. So a protein comes up, copies the DNA, sends the copy into the cell. That's the working blueprint to make the protein. And once I have the copy, I don't need the gene anymore. So guess what happens? Once the RNA is in the cell, the signal disconnects from the chromosomal proteins, the DNA is covered up. And you say, wow, that's really interesting. I go, yeah, you know, for 50 years we've been worshiping at the shrine of DNA, that it controls all our lives. So in this particular case, let's review that again and see how much the DNA is actually doing. So let's watch it. Here comes a signal from the environment, and it binds to the chromosomal protein, changes shape, and now we expose the DNA and say, okay, DNA, do something. Come on, DNA, do something. Do anything, move, do any damn thing. I don't really care. Do something. The DNA doesn't do anything. It's a blueprint. And all those years we said DNA causes things and all that. There is no activity from DNA. So the whole belief system is totally flawed. It's not the DNA that has anything to do with it. It's the proteins and the environmental signals. And all of a sudden you start to see, we had a complete misunderstanding of the nature of how genes work. Now, I want to tell you this understanding of epigenetics because it's really important. Epigenetics, to many, is a buzzword. I don't want to use it as a buzzword. I need you to understand how it works for a simple reason. It's a revolution in our understanding of how life works. So I'm going to talk about epigenetics. I say, how does epigenetics work? And the answer is this. Environmental signals influence the proteins. and the DNA in the chromosome. So environmental signals affect the chromosome. Well, in two ways. Some environmental signals actually alter the reading of the DNA, which I'll talk about. And how does it do that? It adds what's called a methyl group. And you say, all it adds is a little CH3, carbon, hydrogen 3. I say, yeah, the methyl group controls whether the gene is going to be copied or not copied. So the methylation says, are you going to be able to copy this gene or not copy the gene? So methylation selects which genes are going to be activated. Environmental signals also affect the protein. But when they affect the protein, they alter the reading of the gene. And this becomes profound because it's actually the regulation of the DNA is the protein, not the DNA. So I say, well, how does this really work? And I say, first of all, recognize this. You got two sets of chromosomes, one from your mother, one from your father. Each chromosome generally has two genes, well, one gene for each trait, but then you have a mother and a father chromosome, so that means every trait, let's say 7 genes, A, B, C, D, E, F, G. I have 2 A's, 2 B's, 2 C's, etc., one from the mother, one from the father. Well, the interesting point is, which one am I going to read? Because in almost all cases, you only read one of the two. So how do I know which one to read? And the answer is, information from epigenetics involves a process adding CH3 methyl groups, which generally silence the gene. So when I add the methyl groups and select specific genes, those genes are not going to be read. So what are the available genes after the methylation process? One of each. And which ones are selected are not random. They're based on how the mother is experiencing life before her egg is mature and how the father is experiencing life before the sperm is mature. Why? The controls on selecting which genes are actually implied just at the moment of the maturation of the egg and the sperm. They reflect what's going on in the environment. So nature's not stupid just saying, hey, randomly pick genes. Nature says this gene would be better in this environment as opposed to this gene. So nature is selecting which of the genes are going to be used, the mothers or the fathers, based on what's going on in the environment at the time. And this is how methylation works. So epigenetics, in one part, selects which genes can be read and which ones can't be read. But the other part affects the proteins, and I want to mention it in this way. This is a very critical part about epigenetics. Epigenetics changes the readout of the gene, but does not change the DNA code. So if you're born with a mutant gene, you'll always have the mutant gene, but you may not have to read it as a mutant gene. So epigenetics changes the readout, but does not change the DNA. I say, how does that do it? And I say, remember the old days when television wasn't on 24 hours a day? And I say, this is a test pattern. So this is a pattern, like a gene. And it's coming from the broadcast station. And I say, well, the pattern, the gene, is coming from this broadcast. But I say, but if I mess with the dials on the television set, I can alter the shape of the pattern. Here's one form, altering that pattern. I can alter it again by changing the dials on the television set. My most important question is, yes, I changed the expression of the pattern, but functionally, did I change the broadcast? Did I change the original pattern? The answer is no. It's still the same original pattern, but I can change the readout. I said, well, how does that work in our biology? I go, here's a strand of DNA. The loop part is the blueprint, a gene, to make a protein. I say, what about all that extra stuff around there that's not part of the gene? Well, euphemistically, unaware people have referred to this as junk DNA. They say, oh, that's just extra DNA. That's really nothing. A cell is so stupid, it's carried around 1 billion years extra junk. And it turns out, what is the junk DNA? And the answer is this. Built into that DNA strand are protein modules, little clusters of proteins. And guess what these protein modules are? They are the equivalent of the dials on the television set. One is on and off. One is volume. One is contrast, focus, color. And so you can change the readout of the gene by affecting these protein modules. They are, in a sense, really the dials on a television set. So the junk DNA is not junk. It is the most profoundly important DNA of all. Listen to this. The genes get mutated regularly. The junk DNA does not get mutated. It keeps itself very constant. Why? You are getting your pattern of who you are from the junk DNA and getting the parts from the gene DNA. So epigenetics can influence either the gene or the protein modules. So when it alters the protein modules, that's protein regulation, turning the dials and changing the readout. And when it affects the gene, that's the methylation process that says whether you can read it or not. So epigenetics says which genes are going to be read and how they're going to be read. And you say, well, what do you mean I can change the readout? So I say, look, DNA is a blueprint like, and there's some of us that still remember ticker tape machines where a whole message is written on a single tape like letters after letters. That's equivalent of a gene with message code along the tape. So let's say we look at the readout of a gene called the reading gene. So this is the ticker tape of a reading gene, let's say. I say, well, what does epigenetics do? Well, I say epigenetics can alter that reading gene by cutting and pasting. So I can cut off part and make the read gene. I can cut off part, make the ding gene. I could cut off parts and make the add gene. And every time I'm doing that, I'm using the same blueprint, but I'm creating a new protein out of it. So what's the point? And the issue is simply this. For every gene in your body, using epigenetics, I can create greater than 3,000 different versions of the same protein. Some of those versions are healthy versions, and some of those versions are the equivalent of mutant versions. So, as a matter of fact, you don't have to have a mutant gene. You could have a mutant readout. Epigenetics could take a normal gene and turn it into a gene that reads as a cancer gene. And that's why we're beginning to find out a very important point. Cancer is only about 10% connected to heredity, 90% connected to the environment and your response. And the relevance, as you're quite aware, is simply this. If the signals from the environment are controlling the genetics, then I'm not a victim of my genes. If you wanted to say you were a victim of your environment, you could say that, but that's not true because of a simple reason. You can change the environment. You can change your response to life. You can change all of these. All of a sudden, the concept that you are a victim of your DNA is completely false. You are the master of your DNA. It's your perceptions that send signals to the genes. It's your emotions that send signals to the genes. It's the environment that you live in sends signals to the genes. And all of a sudden you realize, yeah, but I can change my emotions, I can change my perceptions, I can change my environment, and all of a sudden you realize, yeah, you are not restricted to a specific gene readout. You are free to change anything in your life. And this becomes very critical because you have to have self-empowerment. And Evan up here was a great example of self-empowerment. Okay, your body's now screwed up. What are you going to do about it? Self-empowerment. I'm not going to stop. I'm going to control my biology with my wishes, desires, and beliefs. And he did, and that's a great example of changing your life. So I go back and I say, what about that central dogma? DNA goes to RNA, goes to proteins. In all the textbooks, I go, yes, but they left out some pieces that are critical. What'd they leave out? Well, what about the chromosomal proteins, the regulatory proteins? Yeah, they control the reading of the DNA. Oh, you left them out of the story. And I go, yeah, but what controls the regulatory proteins? And the answer is the environmental signals. And all of a sudden, you don't live by the primacy of DNA. You actually are representing the primacy of the environment. And all of a sudden, it becomes very important for you to understand that you change the environmental signals, you change the expression of the protein at the bottom. Okay? So basically, I go back to my tissue culture study and I say, yeah, I put cells in a petri dish, change the environment, and I can change the fate of the cells, changing the culture medium. You say, yeah, but that's cells in a plastic dish, Lipton. What about cells in me? I go, well, here's the interesting fact about you. You see yourself as a single living entity, but the truth is, no, you are actually you're a skin-covered petri dish. You got 50 trillion cells inside your body. How does it work? And the answer is simply this. The cells respond to the culture medium. I go, what's the culture medium in your body? The answer is the blood. So I say, so if you change the chemical composition of the blood, it's the same as changing the culture medium in a plastic dish? I go, absolutely. To a cell, it doesn't make a difference if it's in a skin dish or a plastic dish. It's still going to respond to the environmental signal. I say, so the blood in your body is the culture medium? I go, yeah. And I say, what controls the chemistry of that culture medium? Because if you change the chemistry, you change the fate of the cells. I go, well, you read environmental signals with what? Their nervous system. It's perception. I perceive what's going on in the environment. But that's not the end of it, because the next level of control is more important. It's not just the nervous system response to stimulus response. It involves the mind. And why is the mind relevant? Mind is interpretation. And the significance of this, two people can be in the exact same environment, having the exact same influence of environmental signals, but with two different perceptions of what's going on, they have two different totally responses in their body. So all of a sudden, it's not the environment, it's your perception and your interpretation of the environment, which is the mind's role. And when the mind interprets, what it does, it sends signals back into the brain, and the brain releases chemicals that are matching the interpretation. And those chemicals are sent into the blood, or those chemicals are sent via the nervous system to the cells, and the relevance is at the cells. The signals, just like in a plastic dish, the signals control behavior and gene activity. And all of a sudden you see what's at the top of this whole thing is the mind. I go, well, what chemicals should the mind release? Well, I say, well, I was a cell biologist, but we are all, every one of us, self-biologists. And we are creating our biology. As it shows, the brain is creating the culture medium, the blood, and culture medium is being used to feed the 50 trillion cells in your body. I say, well, what's relevant? I say this, you open your eyes, you see someone you love. Oh, when you see someone you love, what kind of chemicals go in the blood? Dopamine, oxytocin, vasopressin, growth hormone. I say, why are those chemicals relevant? I can tell you a simple fact. If I take those chemicals, add them to my plastic petri dish of cells, the cells grow exuberantly well. I say, isn't that interesting? When people fall in love, they're so healthy they glow. Why is that? And the answer is the chemistry in their body is fulfilling the fullest growth potential of your cells in love. I say, well, what about the same person? They open their eyes, but they don't see someone they love. They see something that scares them. I say, oh, fear, that releases a completely different set of chemicals. When you're experiencing or perceiving, interpreting fear, you release stress hormones and inflammatory agents. I say, why relevant? I say, take the stress hormones, inflammatory agents, add them to the petri dish and plastic petri dish with cells, and guess what? The cells stop growing. And that's why fear kills. It's an effort of the system to conserve energy. So don't grow if you have to run away from the tiger. Stop growing and use that energy for escape. Well, it was only meant in history to run away from the saber-toothed tiger. Meaning, okay, you got 20 minutes, scare the hell out of you, and you get away from that tiger, and then guess what? You relax and you go back into growth again. But we don't have that in our world today. We have 24/7, 365 stress. That means every day you read the paper, you watch the TV, you do whatever you're doing. What are you doing? Releasing stress hormones into your body. From your what? Interpretation. Same body, you could look at something and be in love and have a completely different vision. And all of a sudden you realize you are the master, but we have let other people control our interpretations. And as a result, their interpretations control our lives. And we have been disempowered by that. And the interesting understanding of epigenetics has now led into this understanding of identical twins. It was always believed that identical twins coming from the same genes and the same selected genes in the chromosome after conception, that they would be exactly the same forever. And the fact is, this turns out not to be true. That if you look at the genetic readout of identical twins 1 year, 5 years, 10 years, and 20 years after birth, you will find that every year of life, the genetic readout diverges between the two identical twins based on what? Their personal life experiences. And all of a sudden, why this is relevant, because there's been tremendous amount of twin studies to try to prove genetic connectivity when it turns out, hey, even twins change their genetics based on their individual life experiences. So you are not locked in your genes. You change them on a day-by-day basis as you move through life. You could be born with a mutation like the mouse on the left, which is called an agouti mutant. Obviously, it's obese. It's got different color fur, it's got cardiovascular disease, and it's got diabetes, and it dies very young. The mouse on the right is a normal-looking mouse, but it turns out also to be a mutant. I say, how can it be a mutant with a dominant agouti mutant gene, and it looks normal? And the answer is this. In a window during development, the environment of the mother was changed by feeding her some of these nutrients listed in the right corner. At a very specific window of development, the environment changed, and the embryo developing the environment, that signaling from those elements covered up the mutant gene. So you can have a mutant gene and not express it if you do it, change the environment and the entire process. More recently, in San Francisco area, a wonderful researcher, Dean Ornish, took a group of prostate cancer patients and split them up into two groups, one getting conventional treatment with pharmaceuticals, the other group no pharmaceutical treatment. What he did is he helped them change lifestyle, helped them change their diet, gave them stress reduction techniques, taught them how to meditate, no pharmaceutical changes. What happened? 90 days, 500 genes changed their function just by changing that aspect of their lifestyle. And the genes that were affected were mostly the ones affecting prostate cancer. So basically it says, again, the cancer isn't the cause, the initial cause. The cancer is a consequence of a disturbance in your perception of the environment and your programming. That's where it comes from. And it's really wonderful to be up here for a very simple reason. My book came out in 2005. IONS was there with me 22 years ago saying, yeah, this looked like a good idea. Thank you for your support and your encouragement, because in the last 2 years, there's a new science. Behavioral epigenetics, how nurture shapes nature. It says in the fine print underneath, experience and social environment have a role, probably a key role in development. And this is what it's all about. Basically it says this: you are a master of your biology. You control your genetics. But if you don't believe it, then you're going to buy somebody else's belief. So the concept is that you can get a medical diagnosis. You look at the doctor, in this case as a professional, who is diagnosing from the curriculum that he learned. And the significance, if he says you're going to die in 3 months, guess what? You will die in approximately 3 months, not because you needed to die, but because you bought the belief that you will die. And your programming will then, through epigenetics, shorten your life and you will die in 3 months. And the relevance about that is we must return the power back to the individuals, because we are more powerful than a genetic machine. We are the creator of this life. And when you get into the understanding of this and you look at those switches on the surface, the most important part about that, and you heard from Eben Alexander and others that are talking about life experiences after death, and it shows that there are receptors on your cell for, guess what? Your personal identity. And why is that important? No two people have the same cells, because we each have a different set of antennas. Some of these antennas are called self-receptors, receivers of self. Well, if the antennas are receiving self, where are they? They're at the surface of the cell. And why is that important? The outer surface? Because where the heck do you think the signal of self is coming from? It's not in here. You are responding to an environmental signal. You are programming yourself with an information source from the outside that is there forever. The cells come and go, the bodies come and go, the environmental signals of self are always there. That environmental signal, spirit. And this is the whole idea of what IONS has been about for the last 40 years. Congratulations for being the leadership in our world to promote this idea and encourage the change in the population. You were right, and I so appreciate your support for the last 22 years. Thank you very much. Thank you. Thank you very much. Thank you. Thank you very much. Thank you. Thank you. I so appreciate it. Thank you.